My dear student, colleagues, and all the viewers who are watching this program live from Facebook page, I'd like to welcome you all to our International Physics webinar. Today is our 271 International Physics webinar, and we have with us here today Dr. Bradley J. Civic, Associate Professor, Canada Research Chair in Ultrafast Science, Tier 2, and Chemistry and Physics, McGill University, and he has already connected with us. So I'd like to welcome our speaker, sir. Good morning to your part and good evening to yours. Thanks for joining with us and thanks for accepting our invitation. I think this is a very great opportunity for a student to interact with you. Sir, you can start your session.
Great. So thanks very much, Pretam. Thanks for the introduction. It's a real pleasure to be here. And so, before this went live, Pretam and I were talking about exchanging COVID stories. And of course, COVID is in general a very negative thing and has impacted all of us. But these sorts of the availability of these online seminars and being able to meet with and talk with communities that maybe we wouldn't or I wouldn't normally have an opportunity to discuss with is I guess one of the silver linings in it all. So I was really pleased to accept the invitation and have a chance to talk to you about some science that I'm extremely excited about, and also get your feedback on it and discuss with you. And so my seminar title is called "Shining New Light on Material Properties." Okay, so it's talking about a new experimental technique that allows us to really look into condensed matter systems, to look into materials on their natural length and time scales simultaneously, to watch processes unfold at the atomic level and what turns out to be the ultrafast time scale, kind of in real time, to be able to watch these things happen. It's in some sense allows for the experimental realization of a molecular movie or a molecular dynamics type of simulation. That's the quality of information that we're shooting for, and in many instances actually obtain with the new techniques that I'm going to be talking to you about. Okay, so what kind of physics are we talking about today? We're talking about materials physics, all of the physics that happens inside of materials, and on the experimental end, I'm going to talk to you about really very new experimental techniques, some very new instruments that in part were developed and designed by my research group at McGill, but there's a wide community of researchers in this area now, and really there's a very exciting ferment. Lots of very rapid progress, lots of very exciting things are happening in this field, and I hope to communicate some of that to you today. So the subtitle is Structure and Dynamics with Ultrafast Electron Microscopes. So now, and you're actually getting some pictures of the lab here on this slide. So I was told by Pretam that the kind of default audience member, if I can call them that, is somebody doing their bachelor's in physics. And so I've tried to construct my seminar today with that in mind. We're going to, by the end of it, to get to some pretty advanced topics, but I hope to lead you all step by step through sort of an understanding of how these techniques work, what kind of information about materials we can obtain from them and why, and so by the end of the seminar, you should have a pretty good flavor for the kinds of experiments we do and the kind of scope and possibilities that these experiments provide. Okay, so let's I have to start somewhere, and where I'm going to start is perhaps kind of familiar territory to many of you, and that's to describe, well, what's an ultrafast electron microscope, and there I'll start with a conventional electron microscope. And so I think many of you, or most of you, will know that conventional electron microscopy is a very mature field. The technology is pretty mature, and importantly, modern material science is almost unimaginable without the electron microscope and the kind of information that it provides. So a conventional electron microscope uses a continuous electron beam to illuminate a specimen, which has to be quite thin. okay, because it has to be electron beam transparent. The energy of this electron beam tends to be between 100 and 300 kilovolts. Okay, so a lot of the apparatus in here that you see, this cable is a high voltage cable that basically charges up the accelerator region of the microscope. And you have lenses that allow you to focus the electron beam and re-image the scattered electrons onto a detector. And in modern conventional electron microscopes, you can make atomic resolution images of the specimens that you see. You can literally see atoms and their relative positions in materials. But you can also, through a whole variety of different interactions between these high energy electrons and the specimen, you can learn all sorts of things about the nature of the atomic species that make up the sample, right? So, which atom are they on the periodic table? What is their valence state? All sorts of things. It's important to keep in mind that an electron microscope is not only an instrument for making beautiful images of materials. An electron microscope, a modern one, is really a signal generating engine that tells you an enormous amount about materials through the various ways that this high energy electron beam interacts with your material specimen. And the primary ways that we learn about materials in electron microscopes is through the transmitted electrons, okay, the electrons that are transmitted through the sample, and those that get elastically scattered or inelastically scattered. So the elastically scattered ones tell you, okay, essentially they are often used in imaging and diffraction. They give you reliable information about the lattice spacing and atomic arrangements. And the inelastically scattered electrons tell you about the nature of the excitations that are present in the material. And so that's an enormous amount of information, which we hope to be able to build on. Okay, so as I said, electron microscopy is a mature field. A modern electron microscope is a remarkably capable instrument in the laboratory to do materials research. And the current generation of what are known as aberration-corrected electron microscopes, where the lenses are corrected for all their various aberrations. So they can have a spatial resolution well below 1 angstrom. Okay, so the current world record is in fact below 0.5 angstroms. That's approximately the size of a hydrogen atom. Okay, so you can see immediately why you can actually see atoms in one of these instruments. So, but we perhaps, you might say, have reached the end of the race for spatial resolution, further improvements in spatial resolution in electron microscopy. There's no reason to go too far below the interatomic spacing with spatial resolution. So if spatial resolution has run its course as the main driver for the development of electron microscopy instrumentation, because we've reached the end game there, what are the new frontiers for electron microscopy instrumentation? And I would say they're essentially two primary avenues. There's one that provides you improved or differential contrast across the sample, being able to distinguish grain boundaries from bulk. You can to determine which atomic species make up both, and so there's a lot of work on what's known as EELS, that's electron energy loss spectroscopy, that makes use of the inelastically scattered electrons that I showed on the previous slide. Okay, but the other axis of electron microscopy development is to enable new kinds of experiments inside of electron microscope instruments. So called in situ and in operando investigations, okay, that allow you access to much higher time resolution than is available in a conventional electron microscope, or, okay, to provide tunable environments in which to watch a sample's response to stimulus. Okay, so the talk I'm giving you today really explores this this second axis for the development of electron microscopy. That is, conventional electron microscopes have been doing a very good job of telling us about the static or equilibrium structure of materials and their materials are dynamic species. There are all sorts of processes in materials that lead to changes in structure. There are dynamic processes in materials, even at equilibrium, that control the properties of those materials. And conventional electron microscopy has had a very difficult time interrogating any of those. Okay, so a really important axis for the development of electron microscopy is to increase the time resolution to be able to get a direct view of these fast processes and materials that have been out of reach of conventional electron microscopy up until now. So that's to develop electron-based methods with ultra-fast time resolution. Okay, for the sake of this presentation, that's defined as time resolution between the femtosecond, 10 to the minus 15 of a second, to the nanosecond, 10 to the minus 9. Okay, to be able to watch structures in materials change on these timescales is really a major new frontier that impacts on physics, chemistry, and material science, and biology, actually, if I wanted to add that here. So pretty much these kinds of developments in electron microscope technology can be expected to touch on all of the traditional scientific disciplines, because the goal, as stated here, is clear and important for all of these fields, right? That is, to watch processes as they occur in materials on both their natural length and time scales. Okay, and so I'll talk a little bit about that to bring everybody on side, and is to make molecular movies, right? So what are the natural length and time scales of processes that occur in materials? So processes that occur in materials lead or result from often rearrangements of atoms in space, right? So that sets both a natural length and a natural time scale for processes in materials, right? Atomic rearrangements. Okay, so on what length scale do they occur, and on what time scale do they occur? Well, moving atoms takes some time, but not very much. Okay, so the making and breaking of chemical bonds and molecules and materials typically occurs on a time scale of 10 to the minus 13 seconds. So that's 100 femtoseconds. Where does that number come from, and how should you think about that? Okay, so atoms inside of materials are limited in how fast they could move, right? So the speed of sound is a good number. So the atoms a neighboring atom will feel the displacement of its neighbor, right? After a delay time related to the speed of sound in that material, right? So take a specific atom, move it, displace it from its position. How is that information communicated through the rest of the solid? Well, the speed of sound is the operable time scale there, right? So how far does that information have to travel between sort of an atom and its neighbor? Well, the answer there is, on the order of an angstrom, right? Because in materials, the interatomic separations are measured in angstroms, 10 to the minus 10 of a meter. So these distances are very small, right? The speed of sound, okay, 10 to the minus 10 of the meter. So the distances are, okay, speed of sound is typically 1,000 meters per second in many materials. Okay, the distances between atoms are 10 to the minus 10 of a meter. Okay, so what's the time scale, right? It's the distance divided by the velocity, or the velocity, distance divided by the velocity, and you get a typical time scale of 10 to the minus 13 of a second. Okay, so this is fundamental, right? This is this time scale is fundamental in materials. Okay, we can make the bold statement that all atomic motion that takes place inside of molecules and materials is essentially frozen in place if you can make direct observations of those arrangements inside of a time scale less than or around 1 femtosecond, 10 to the minus 15 of a second. And that's quite a remarkable thing to say, right? So that if I can go in and measure the structure of a material, okay, with a flash or some other means, and I can make that measurement on a time scale that's on the order of 1 femtosecond, okay, the atoms are frozen in place. They're not moving, okay? And if you can do that, then you can watch whatever atomic rearrangement process you're interested in unfold in time, because you can see how the atomic trajectories are moving as a function of time, because you can capture all of the details of that process. And so that's one of the ideas here. One of the sort of unifying themes of this research is to be able to do that, okay? To be able to make observations on materials that are fast enough to be able to freeze atomic positions in place and watch these processes occur in real time. So, and you can immediately see, well, the spatial resolution challenge is clear. I think everyone, you have to be able to see atoms, or some approximation of that, but you need to be able to see atoms also move on roughly the 1 femtosecond or to 100 femtosecond time scale. So, and a femtosecond is 1/millionth of 1/billionth of a second. So the experimental challenge here is severe. So how do we do it? How do we sort of construct an instrument that's capable of not only resolving atoms, but being able to watch them move? And that is to combine the electron microscopes that I spoke about up until now with another piece of technology, which is femtosecond lasers. So maybe some in the audience will know that in 2018, the Nobel Prize in Physics was awarded to Donna Strickland and Gerard Moreau for their development of a critical piece of technology, which is called chirped pulse amplification. Okay, you can actually see that inside of this box if you're an expert, but I'm not going to have time to speak about that bit of technology specifically, other than to say it's allowed for the development of very capable, both powerful and stable lasers that work rather differently than the ones you might be familiar with in your everyday life. That is a laser pointer, okay? So laser pointers generally produce a continuous stream of light output when you push the button. This class of femtosecond lasers instead produces, rather than a continuous beam, a pulsed beam output, okay, where the laser pulses come out at some repetition rate, say 1,000 pulses per second, but each pulse is itself extremely short, okay? So measured from front to back, these pulses can get down below 15 femtoseconds, actually all the way down to a handful of femtoseconds, okay? So a 15 femtosecond pulse only contains so a 30 femtosecond pulse contains in general about 10 oscillations of the electromagnetic field at 800 nanometers. Okay, so these are true and genuine pulses of light, where you have a countable number of electromagnetic field oscillations underneath the envelope of the laser pulse. Now, so this does a couple of things for you. The important thing for my talk today is that this short duration burst of light provides you access to that important time scale in materials whereby these processes are occurring. Okay, so let's move on. Okay, so a critical piece of the puzzle is are these short laser pulses that can be produced by modern femtosecond lasers. Okay, and so here's a plot that shows you as a function of space and time where a set of important materials processes occur. So the important thing to note is that up in this corner, right? So over here, you have processes that are occurring, near the atomic scale and are occurring on what I defined as the ultrafast time scale between 10 to the minus 9 seconds and 10 to the minus 15 of second. And this involves the making and breaking of chemical bonds, the making and breaking of bonds inside of materials, as I indicated before. Okay, it involves important processes like melting and resolidification. There's nucleation and growth in materials that overlaps with this range of timescales. Phase transitions in materials occur on this timescale, and there's other processes which the experts will know, but maybe the people halfway through their bachelor's in physics will not heard of yet. But the coupling between electrons and lattice vibrations, okay, otherwise known as phonons in materials, are extremely important to an understanding of material properties. And the natural length and time scales of this electron-phonon coupling, which determines many important things like both thermal and carrier transport in materials. Actually, conventional superconductivity emerges as a result of electron-phonon coupling in materials. A huge range of very important materials phenomena directly occur due to this electron-phonon coupling. And so we'd like to be able to access this range of phenomena reliably with an experimental technique, and it hasn't been possible to do that until recently through the development of the methods that I'm talking to you about today. Okay, so how do we do that? How do we access this range, this critical range of phenomena in materials with the tools that I presented to you so far today? Okay, and this I present to you as my the Civic research group, or the Civic group research equation. Okay, so this is how our experiments come together. We take femtosecond lasers, which, as I indicated, provide us access to the time resolution required to perform our experiments. We combine those in novel ways with electron microscopes. Now, this is shown here, a conventional electron microscope. In my lab, we actually have to design our own electron microscopes, as I'll show. But if you combine the time resolution of femtosecond lasers with the spatial resolution of electron microscopes, okay, you actually have the ability to make molecular movies, to be able to watch the course of atoms during at least photoinduced processes in materials to quite some level of sophistication. Okay, so I'll talk a little bit. I want to talk a little bit about that, how that's done. Okay, so how do we do that, and what are the challenges? But that's the if you can follow sort of this research equation, you can follow most of what remains in my talk here. Okay, so as indicated in the previous diagram, that allows you to follow materials under extreme conditions where atomic positions are varying rapidly as a function of time. So that allows you access to understanding phase transitions that are occurring not close to equilibrium, where you normally view such things, but very far from equilibrium, strongly driven systems. And importantly, in an example that I'm going to show you in a minute, it also allows you to do entirely new kinds of experiments. You can actually probe how optical excitation of materials, how coupling materials to the external stimulus of light allows you to generate new properties in materials that can't be obtained at or near or under equilibrium conditions. So, and if you want a picture to understand what that means or how that works, here's such a picture. Okay, so here we have a potential energy, a free energy diagram for some complex material, and at equilibrium, the phase that you see in the material is generally the minimum on this free energy surface. Okay, and so there is some order, okay, that is critical to understanding sort of the structure of that equilibrium phase, and this is what you see at equilibrium. But that doesn't mean there aren't many other minima that might be present somewhere else on this free energy surface. Okay, and that these other minima on this free energy surface might yield materials with properties and structure, okay, that could be very, very different than the observed properties of the equilibrium phase that's associated with this one minimum on this otherwise very complex potential energy surface. So how might you be able to sample these other minima and simultaneously be able to determine what's the nature of the material, at those other minima where you have different orders and properties? And so the one experiment that you can do to kick the material out of this equilibrium phase and sample the rest of this potential energy diagram is to shine laser light on it. Okay, that excites the material, okay, higher onto the potential energy surface, okay? And if you're able to follow the evolution of the material, both its structure and its properties, you can actually follow the material as it comes down on this potential energy surface and potentially occupies one of these other minima, where you have a metal stable phase, maybe short-lived, maybe actually quite long-lived, where the material is exhibiting a completely different structure in terms of whatever order is being measured here. Okay, that could be a magnetic structure or a crystallographic structure or a charge order. There's many different sort of possible orders that we could be talking about here. And this material has very different properties and structure than the material over here. And in really interesting cases, you can kick the material over to a competing ground state that is inaccessible because of the large barrier here, that is inaccessible by equilibrium means. But this non-equilibrium photoexcitation provides you an ability to kick the material into this competing ground state. Okay, and so if you want to watch this happen, okay, you can use the tools that I talked about on previous slides. Okay, so you can watch the material sample these different minima by using an electron microscope and determining the structure of the material as it moves along this coordinate. Okay, and probably the most exciting example of research of this kind that I can give that has been demonstrated comes from Andrea Cavallari's group at the Max Planck Institute for Structural Dynamics in Hamburg, where they have investigated unconventional, non-conventional superconducting materials, okay, and have demonstrated that through optical excitation, you can enhance superconductivity at temperatures that are even above the superconducting transition in the material. So this is a very interesting and exciting avenue for further research, where we can potentially access materials and properties that nature will not provide us naturally at equilibrium through, under non-equilibrium conditions. Okay, so let's talk about Okay, so that's kind of the end of the long introduction to bring everybody on side, and now I want to get into some of the nitty-gritty, some of the details. We're going to start talking about what it is that you are seeing when you interrogate a material with an electron beam. Okay, so I'm going to start with the simplest and most elementary idea. Okay, and that is, okay, if I have a single isolated atom floating in free space, okay, and I subject that atom to sort of an intense or a high voltage electron beam that then sees that atom, what is it that the electron beam is actually seeing? Okay, and the answer to that at the end is that this localized, the atom presents a localized charge distribution, right? The nucleus, positively charged nucleus at the center of the atom, surrounded by this quantum mechanical electron cloud, right? That's negatively charged, right? So that localized charge distribution that involves both positive and negative elements presents an electrostatic potential, okay, to the incident electron beam, okay? And that electrostatic potential can be varying in time if the position of the nucleus changes, or the nature of the surrounding electron cloud changes as a function of time. And what you're seeing here is an equation that I think many of you in the audience will have seen in an earlier undergraduate course. This is Laplace's, the Poisson equation, okay, for the electrostatic potential of a given charge distribution. And the charge distribution for this atom here is, of course, made up of the nucleus, which is well approximated as a delta function at the center. And this what is generally relatively complicated surrounding charge distribution, which for an isolated atom is going to be spherical, but for an atom inside of a material is definitely not spherical, right? So when bonding electrons are included in here, the electronic distribution can actually be distinctly non-spherical, okay? So the answer to the question, what is it that this electron beam is probing in materials, is that the electrons are scattering or probing the localized charge distribution presented by the atoms, okay? So how do we understand the scattering, okay? So we have electron beam incident on our material, okay, that made up of atoms, these localized charge distributions, okay, that present this electrostatic potential to the incident electron beam. Okay, so what do you measure on the other side of the sample from the perspective of the electron beam? Well, you measure, in the simplest cases, elastically scattered electrons. So the electrons see this potential and scatter off of it. And what you measure is the intensity distribution of those scattered electrons. So for a single atom, and the distribution of scattered intensity as a function of momentum transfer is well described by something called the first Born approximation, okay, which I think there'll be many in the audience who have seen before in a course. Okay, the distribution of scattered intensity is proportional to the Fourier transform of the spatial distribution of the scattering potential. Okay, and so for a single atom, that's pretty simple. So if you have if the electrostatic distribution can be described as a Gaussian, this scattering intensity is also described by a Gaussian, where you have a reciprocal kind of Fourier transform relationship, right? So the more localized, the more delta function like the scattering potential, the larger a range of scattering angles that are seen on the other side, and vice versa. Okay, so if you have a single crystal, if you no longer have just a single atom, but you have a periodic arrangement of atoms, you can actually express the electrostatic potential as essentially a Fourier series, right? So this electrostatic potential is described by a Fourier series where you have amplitude coefficients and you have spatial frequencies that define the periodic arrangement of the atoms. So if I put this into here, what you get is this expression. And I think many undergraduates in physics will recognize that this integral over all space, okay, this complex exponential is the equivalent of a delta function in that case, and it sifts out only when Q is equal to H. And so the simplification here that comes from an infinite periodic arrangement of atoms is that the scattering intensity is proportional to this set of delta functions, where this delta function is only non-zero when the scattering vector direction is equal to one of the reciprocal lattice vectors, one of the fundamental periodicities that are present inside of the crystal, which is why if you have a single crystal in front of an electron beam, the pattern of scattered electrons looks like this, right? So you have a spot-like diffraction pattern, okay, which is well known in crystallographic investigations of single crystal materials, right? This pattern of spots, okay, is a manifestation of what's known as the Laue condition for crystallography. That is, you only have non-zero scattering of electrons or X-rays when the scattering vector direction, here Q, is equal to one of the reciprocal lattice vectors, one of the fundamental periodicities of the crystal that one is interrogating. And even more than that, okay, the amplitude of that peak, okay, is actually directly related to the Fourier expansion coefficient in this series, right? The stronger you have a modulation of the electrostatic potential at that spatial frequency, the brighter will be the diffraction peak in the scattering pattern. And so this kind of diffraction experiment provides you very important atomic level details about the nature of the electrostatic potential that is presented by the periodic arrangement of atoms. Okay, so that's some fundamentals. Right? Normally, if you kind of go to a crystallography textbook, you don't see what I just showed you on the previous page. What you see instead, okay, is a description for that factor A of H, which is given, okay, in an atomic basis. It's given as the sum over all of the atoms inside of the unit cell that make up the three-dimensional crystal structure. Okay, so that A of H is expanded into an atomic basis where you sum over all of the atoms inside of the unit cell. Okay, and there are three factors inside of this sum. The complex exponential involves the scattering direction Q and also the relative position of all of the atoms inside of the unit cell. Okay, so there's a complex phase which shows you that what an electron diffraction or an X-ray diffraction pattern is really a interference pattern of scattering from all of the atoms that make up the unit cell. Okay, so there's this complex phase which depends on the scattering direction and the relative positions of the atoms inside of the unit cell. You have a scattering factor, which is atomic species dependent. Okay, heavier atoms have a stronger scattering factor, and lighter atoms have a weaker scattering factor. And then there's a third term in here, which is called the Debye-Waller effect, and it's related to the random thermal motion of the atoms about their lattice sites. Okay, but okay, so you can. Coming back to the discussion I gave before, you've got X-ray or electron scattering, right? Which generally tells you, in the conventional case, tells you about the equilibrium positions of the atoms, right? But for a dynamic materials process, right, the positions of the atoms can change as a function of time, okay? So if the positions of atoms are changing as a function of time, that modulates this complex exponential and therefore also modulates the measured scattering intensity, the intensity of these peaks, right? So there could be charge transfer. Okay, these atomic scattering factors can also change in magnitude. That also is going to have an effect on the measured scattering intensity. And of course, the random displacements of atoms about their lattice sites can also change. And so this factor can also change. So for dynamic materials processes, all of the terms that make up the amplitude of scattering at this particular point can change as a function of time. But you can also have phase transitions that change the periodicity of the crystal, right? You can go from one crystallographic phase to another, where the fundamental periodicities of the lattice change. You have a different crystallographic symmetry in the one phase than the other. These H's can change, which actually lead to the decrease of some Bragg peaks and the increase of Bragg peaks that are not yet seen inside of this diffraction pattern. So there's an enormous range of information that one can obtain about dynamic materials processes by scattering of electron beams off of crystals, okay? And that information can be read out as modulations in the intensity of the diffraction peaks, and also the decrease in intensity of some diffraction peaks and the emergence of new diffraction peaks. Pretty much everything that you would want to measure about the lattice structure of a material is directly obtainable through an experiment like this. So the name of the game in ultrafast electron diffraction has been to measure. Sorry about that. I included a so you want to measure the intensity of Bragg peaks as a function of time during material processes. And so how do you do that? So how does the whole experiment come together? And that's shown schematically on this slide. So in general, these experiments work by what's known as a pump probe approach, okay, where you have the material, the sample that you're interested in studying, and you subject that material to laser excitation, which in general drives vertical excitations of electrons in the material. So you, in general, use the laser pulse to drive the electrons out of equilibrium. You excite specific photochemical transformation in the sample, okay? But laser pulses can also be used to directly modulate interatomic distances through direct coupling to vibrations, okay? So laser pulses can be used to either excite electrons in the sample or directly drive vibrations in the sample and probe different kinds of processes that might be happening in materials, okay? So the essential parts of these experiments is you have your laser excitation, which drives the material out of equilibrium, okay? And you have a short bunch of electrons that probes the material and obtains an electron diffraction or electron scattering pattern that is representative of the instantaneous atomic arrangement of the atoms in the sample, okay? And so in general, in such an experiment, the time resolution, your ability to follow the atomic rearrangements that are occurring in the sample, are determined fundamentally by the electron pulse duration, okay? What you can say about the nature of those motions is related to the signal to noise, right? And the signal levels in these experiments are generally determined by the electron pulse fluence, the number of electrons that you're able to scatter off of a sample per beam area on a shot-to-shot basis, okay? And so in order to get these experiments to work, you have kind of two simultaneous challenges, okay? You need to produce a pulsed electron beam, okay, that is shorter, okay, in terms of its time when it hits the sample than the underlying atomic level dynamics that one is hoping to measure, okay? But you know, an electron pulse contains a bunch, potentially millions of electrons inside of it, okay? Each one of those electrons carries a charge, okay? The electrons are mutually repelling in this bunch through a process called space charge interaction, right? And so it turns out to be not very easy to produce a short bunch of electrons due to this Coulomb repulsion that's happening inside of the electron pulse. And so one of the technical challenges of getting these experiments to work is squaring these two requirements of the experiments. The ability to produce short pulses at the sample so that you can follow the dynamics, the structural dynamics, and to have those pulses contain a sufficient number of electrons so that you actually can make reliable measurements of those processes. And that has been a technical problem that my research group has worked on actively. Ultrafast electron diffraction and diffuse scattering. Pardon me. Okay, so the overall idea is summarized in this nice set of images that were taken by Edvard Muybridge in 1878. Right, so we're going to use electron scattering to determine kind of atomic spatial resolution pictures of the arrangement of atoms in the material, and be able to follow those arrangements as a function of time during a material process. And so the idea is to use electron scattering the same way that Muybridge used flash photography in order to determine the sequence of postures that a horse moves through as it gallops. And the question that was important at the time that was unanswerable using other techniques was, is there a point in time during a horse's gallop where the horse has all four hooves off of the ground at the same time? And so there are a never-ending range of similar kinds of questions that we have about the nature of atomic scale processes in materials that are just like this. Okay, and in this particular case, flash photography, okay, instantaneous flash photography was able to answer the question, right? If you can actually watch the horse gallop and you're able to take photographic images, okay, that have a sufficient time resolution, okay, so that you can actually resolve the motion of the horse's legs, you can see that there is this moment in time, okay, during a horse's gallop where all four hooves are off of the ground at the same time. And so there's a direct analogy between this, this effectively instantaneous optical imaging modality and the kinds of experiments I was talking to you about electrons and atomic resolution. So the primary difference between what you're seeing on your slide here and the kinds of experiments we can do is both the nature of the spatial resolution and time resolution that's obtainable. Okay, so one of the most difficult things to do is to actually get the required ultra-short electron pulses at the sample. And the difficulty there is what I mentioned a couple of slides ago. So our electron microscopes work through photoemission of the electron beam. So we make use of the pulse train coming out of the femtosecond laser. We shine those laser pulses onto a photocathode in the electron microscope. And every time a laser pulse hits the photocathode, we generate an electron pulse. Okay, and because, as I showed, it's quite straightforward to produce femtosecond laser pulses. At least initially, it's quite straightforward to produce a femtosecond electron pulse. The problem comes when the electron pulse comes out of the photocathode and propagates in the instrument. There, okay, depending on how many electrons you put in each pulse, the Coulomb repulsion inside of the bunch changes the electron pulse duration as it propagates, as shown here through some simulations. Okay, so it's easy to produce through photoemission an ultra-short electron pulse that's only femtoseconds in duration. But when that pulse comes out, what you have is effectively a pancake of charge, where the electrons at the front are being pushed forward by the electrons at the back. The electrons at the back are being pushed backwards by the electrons at the front. And as this pulse propagates, okay, it gets longer and longer in time because of the Coulomb interactions that are taking place inside of this bunch. So this is quite severe. Okay, these are simulations that only show 3,000, 6,000, and 9,000 electrons per pulse. And you can see that already, even with this few number of electrons, by 1 nanosecond of propagation, which is only about 10 centimeters for a 30 kilovolt electron, the electron pulse duration has broadened well outside of the femtosecond regime into the picosecond regime. And so this was seen as a maybe a showstopper at the time for performing truly ultrafast electron diffraction experiments. But the good news is that this space charge broadening of the bunch also leads to other effects, okay, that provide the solution to this space charge broadening problem. Okay, so in addition to this broadening, what you find inside of the electron pulse is that the phase space distribution inside of the bunch changes as a function of time. The photo emission that generates the electron pulse is a statistical process. Okay, so there's no reason that the first electron to be photo emitted should have a different energy than the last electron, but there is a distribution of photoelectron energies. So that's indicated in this figure with these little dots. So each one of these dots is a single classical electron inside of this electron bunch that contains about 9,000 electrons. So there's about 9,000 dots here. So at time equals 0, what you have is a completely uncorrelated blob. Okay, so on this axis, you have the relative on-axis velocity of the electron propagating in the direction of the electron beam. So the relative velocity of the electron, the fast ones are up here, the slow ones are down here. And on this axis, you have the relative position of the electron inside of the bunch. Okay, the electrons at the front of the bunch are over here, and the electrons at the back of the bunch are over here. And you can see what happens to this phase space distribution as the pulse propagates. Okay, by, as I said, about 10 centimeters after photoexcitation, what you have is a beautifully linearly correlated electron bunch. Right, the fast electrons are at the front, the slow electrons are at the back, and all of the other electrons line up on this nice straight line in between. Okay, this is a robust feature of the propagation dynamics of these femtosecond laser-produced electron packets. This happens for free. There are fundamental reasons why this happens. I won't have a chance to go into it, but the important thing for the remainder of my story is that this nicely provides a solution to this space charge broadening problem, provided you can subject this this linearly correlated electron bunch to time-dependent electric fields, which is a technology that my group has developed in these instruments. Okay, so the technology here is called a radio frequency cavity. It's borrowed from the particle accelerator community, and you can make these cavities that have specific resonance frequencies, which have specific modes where the electric fields have given arrangements. What's shown here is called a pillbox cavity, and this is the TM010 mode. Okay, so those are technical details. The important thing is that this electric field inside of this cavity oscillates very rapidly in time. In the cavity we have in my lab, the period of the oscillation is 300 picoseconds. Okay, so if I take one of these space charge broadened electron pulses, which have this linear momentum position correlation that I showed you on the previous slide, and I inject it into this RF cavity at exactly the right point in the sinusoidally oscillating cycle, I can arrange things such that the electrons at the front of the pulse see an electric field that slows those electrons down. Okay, does negative work on the electrons, slows them down. Okay, the electrons in the center of the bunch see no field at all because of the oscillatory cycle of the electric field inside of the cavity. The electrons at the back see an electric field that accelerates the electrons. Okay, if I do that, okay, I can rotate the phase space of this electron pulse from this positively chirped configuration into a negatively chirped configuration. And here's the important thing here, right? So here the fast electrons are at the front, the slow electrons are on the back. This pulse will continue to get longer forever. Whereas this pulse has the fast electrons at the back and the slow electrons at the front. These fast electrons will eventually catch up with the slow electrons, and some point downstream, you have a short pulse again. Okay, and this behavior is tunable. Okay, you're by adjusting the electric field strength in the cavity and the frequency, you can change the temporal focal length of this this element, and what you have is effectively a temporal lens. Okay, for the re-temporal focusing of these space charge broadened pulses, and this provides you access to high time resolution electron scattering instrument that contains many electrons per pulse. So this is a picture of the instrument that my group developed inside of my lab, and its current important figures of merit. So the electron pulse duration at the sample has reached this 100 femtosecond time scale that I told you is critical for following much of this motion. But just as important, okay, the electron pulses contain many electrons per pulse, okay? Upwards of 1 million electrons per pulse, which is an X-ray equivalent flux of 10 to the 11 to 10 to the 12. So we're competitive with latest generation ultrafast electron source user facilities that are becoming available elsewhere. So this is a remarkably powerful instrument. I would say the most powerful of its kind in any research lab worldwide. And we've been having a lot of fun with this, doing all sorts of experiments of the kind that I described to you in my introductory remarks. And so I want to use what remains of my time to show you just a single example of what one can do to probe dynamic processes in materials and uncover phases of materials that don't exist at equilibrium, but you can produce through optical excitation. And the example I'm going to give is how you can use optical excitation to control the structure and properties of a material called vanadium dioxide. It's an absolutely fascinating benchmark problem in materials or condensed matter physics. Okay, vanadium dioxide is what's known as a strongly correlated transition metal oxide, where the electronic properties of the material are not well understood using the elementary band theory of solids. And one of the most interesting parts of the physics of this material is the well-known metal to insulator transition that occurs just above room temperature, around 68 degrees Celsius. Okay, at high temperatures, okay, temperatures higher than 68 degrees Celsius, the structure of the material is high symmetry. Okay, so it has a rutile crystallographic arrangement where the vanadium atoms are nearly perfectly octahedrally coordinated by oxygen atoms, and you have these chains of octahedrally coordinated vanadium atoms that are equally spaced along the rutile C-axis of the crystal. As you cool this material down below 68 degrees Celsius, you get a structural change. There's a crystallographic rearrangement where the vanadium atoms, which are initially equally spaced in the high temperature, high symmetry phase, become dimerized along this rutile C-axis. So you, instead of having equally spaced vanadium atoms, you have dimerized and undimerized vanadium pairs as you move up, where the rather than the vanadium atom being centered in the octahedra, there's also a tilt where these atoms move out of the centers of these octahedra. Okay, and associated with that crystallographic reorganization to what's known as a monoclinic crystal symmetry, there is an enormous increase in the resistivity of the material. You go from the material in this high symmetry phase being a reasonable metal, okay, good conductor of electricity, to an insulator, effectively a semiconductor, which is a very poor conductor of electricity. In good samples, the difference in resistivity can change by about 5 orders of magnitude through this process. And what's remarkable is that this physics, the understanding of how this crystallographic reorganization of the material is related to this dramatic change in electronic properties, has been the active subject of study in physics for many decades with no good answer, because it's such a complex problem. Okay, so the question always has been, to what degree can this change in electronic properties be understood solely as the result of this crystallographic transformation, or how much does more sophisticated questions of quantum materials, otherwise known as sort of a Mott-Hubbard transition, where the electrons localize on lattice sites, how much must that be invoked to understand this change? And so the experiment we did is to actually visualize this metal insulator transition as it occurs in the material and its natural length and time scales using the approaches that I showed you before. Okay, the experiments that we do are twofold. Okay, so we do one of these pump probe electron scattering experiments, which tell us, as I indicated, about the interatomic arrangements of atoms inside of the unit cell. So ultrafast electron diffraction tells us about the instantaneous configuration of the atoms inside of the unit cell. And we can follow that as a function of time after laser excitation, which takes you from the monoclinic phase to some laser excited state on the other side. Okay, and in a parallel experiment, we can actually follow using time-resolved terahertz spectroscopy, something that I'm not going to have a chance to discuss much here, but you can measure the change in electrical conductivity of the sample all the while during this process. And so we have these two direct measurements, both on the femtosecond timescale, that allow you to determine the instantaneous configuration of the unit cell. Is it monoclinic? Is it rutile? Is it something in between? And to be able to instantaneously determine whether that particular phase of the material is behaving like the semiconductor, which is the parent phase, or the metal, which is the phase that one moves into. So I won't dwell on this, but with the instruments we've developed and designed in my lab, can produce data sets that are unprecedented in terms of its signal to noise and fidelity. We can measure the optically induced changes in the intensity of any one of these peaks with a fidelity of better than 1 part in 10 to the 4 or 10 to the 5. Okay, we can measure every single peak inside of the visible window with such fidelity, which allows us to really do what I suggested before, which is to make atomic resolution movies of the optically induced changes in structure that occur in this material. And I'm going to give you a summary of what we find when we do this. So there's 2 papers where these were reported, one shown here and the other one I'll show in a previous slide. All of this complex physics that's shown here essentially distills down into 2 easy to understand processes. There are changes in the structure of the material that occur very fast on timescales less than 500 femtoseconds that we can read out through these changes in diffracted intensity. There are other changes that occur relatively slow on a fluence-dependent 1 to 6 picosecond timescale, and you can see the character of those changes. The character of these slow changes in diffracted intensity are completely unlike the character of the fast changes in diffracted intensity. You don't need any particular training in crystallography to understand that these changes in diffracted intensity must mean a very different structural rearrangement than these changes in diffracted intensity. And so we're able to determine, using these techniques, what is the nature of the structural transition occurring in the material on those fast and slow timescales. And so, as I said, electron diffraction in general directly interrogates the electrostatic crystal potential in a material. So you actually can see the atoms and how strongly they're scattering. And so this is a reconstruction of the electrostatic crystal potential of vanadium dioxide in the monoclinic phase that comes directly out of the data. Okay, you can see these vanadium dimers in this particular slice, and they're dimerized and tilted, just like I said they were. Okay, and these white dots here are the oxygen atoms in that one slice that are octahedrally coordinating each one of these vanadium atoms. So what's the difference between the observed fast process and slow process? Well, the fast process can easily be understood crystallographically. It is to be understood as the laser excitation essentially breaking these vanadium dimer bonds and the atoms moving into the relative arrangement that's to be expected in the rutile structure that I showed you, where you have an equally spaced separation of the atoms along this axis, and you've lost the tilt of these dimers along that same direction. Okay, so the fast process that we're observing is understood as just laser-induced transition between the low temperature equilibrium stable phase, which is this monoclinic structure, and the high temperature equilibrium stable phase, which is this rutile structure. Okay, that's not unexpected, right? If we start in the monoclinic phase, laser initiate a phase transition, this is what we were expecting to see. Okay, the surprise comes based on that slow process. What is the nature of the structural rearrangement associated with the slow dynamics? And that is very different indeed. Okay, so the crystallographic or the structural rearrangement that's associated with the slow process is indicated here. Okay, so you can see this slow rearrangement leaves the vanadium dimers intact. Okay, so you still have dimerization of the vanadium atoms along the rutile C-axis. Okay, those dimers are still tilted with respect to the rutile C-axis. So from the perspective of the vanadium atoms, you still have a monoclinic structure, right? But something important has changed. The oxygen atoms that coordinate the vanadiums, okay, which used to be equivalent, okay, in this particular phase of the material, have now become non-equivalent. These oxygen atoms appear bright, and these oxygen atoms appear dim. So there's been a change in there hasn't been a change in the interatomic separations, but there's been a change in the charge of these various species. There's been a movement of charge away from the metal onto the oxygens, but not onto all of the oxygens, onto a specific oxygen pair. And what you have in this material, evident in this example, is you have an anti-ferroelectric charge order that has been induced through optical excitation along this axis of the crystal. Okay, there is no equilibrium stable phase that has this structure. Optical excitation produces this metastable phase that appears nowhere on the equilibrium phase diagram for the material. And here's the most interesting thing that comes from the partnering of these measurements with the terahertz results. So I showed you that the monoclinic phase of the material at low temperatures is an insulator, it's a semiconductor, and I showed you that the rutile phase of this material at high temperatures is a metal. So you have a crystallographic rearrangement that is associated with an insulator to metal transition. The interesting thing about this phase, which is also monoclinic, but has a different charge order than the equilibrium stable phase, this is also a metal. This, the electrical conductivity of this phase of the material is almost as good as the electrical conductivity of the equilibrium stable rutile phase. So we've shown through these set of experiments that optical excitation of this strongly correlated insulator can yield a new phase of the material that's not present on the equilibrium phase diagram, okay, that combines the properties of both of the equilibrium stable phases. okay, the monoclinic lattice distortion, and metallic conductivity, which is quite remarkable. And that answers a lot of the questions about the fundamental nature of the metal insulator transition in the material. And that's described in sort of this follow-up paper here. So I've used up my time. I'm going to have to go right to the end. I had intended to discuss something, a bit of an advanced topic. This is quite challenging, and maybe it's best that I don't go through it. I'll just say we've moved beyond molecular movies in my lab, directly to being able to visualize the strength of electron-phonon interactions in materials, to be able to use these electron scattering techniques to directly interrogate how strong the interaction is between the carriers, the electric charge carriers in the material, and the lattice vibrations. And there's a range of
scattering, or I0.
There's a range of important measurements that we've done. Some on graphite, which are indicated here. Okay. We've also made measurements on titanium diselenide, which is a transition metal dichalcogenide, which has interrogated the nature of a phonon softening mechanism in this complex material. And we've also very recently reported how these new techniques allow you to directly visualize polaron formation, interesting quasi-particle that can be generated inside of polar lattice environments. We've shown how these techniques provide access to that information as well. Okay, to end, I will say that if anything that I've said today sounds interesting to you and you want to follow up with some further reading, I was involved along with Ilka Arslan at Argonne National Lab in Chicago, and CJ Wang, formerly of the SLAC facility at Stanford, in putting together a special issue of the Materials Research Society Bulletin, the August 2021, so very recently. The August 2021 special issue is called Frontier Non-Equilibrium Materials Science, that is enabled by ultrafast electron methods. There are 6, there's an introductory article and 6 research articles specifically on this theme that are written for a broad material science audience, which can give you a very nice picture, can actually supplement the picture that I provided you today, which only used one very specific example to illustrate these techniques. If you read this special issue, you'll have 6 excellent examples of different different applications of these instruments.
I'd like to acknowledge,
and I will end there. So I will acknowledge my research group, the students, the PhD students that contributed to the work, former PhD students, my collaborators, both at McGill and internationally at Northwestern and Columbia University. And I thank you for your attention.
Yeah. Thank you, sir, for your wonderful presentation. We have got few questions, so if you allow, we can start our discussion session.
Yeah, sure. So should I stop sharing my screen, or should I leave that? Should I leave that up?
You can minimize, or you can stop share. Minimize this space, and then come in StreamYard. Then you can see the question.
All right, I'm here.
The first question is, why does dynamic light scattering provide larger size value than transmission electron microscope?
So I think this is a question about spatial resolution. That's how I'm interpreting that. So well, okay. So there's a couple of answers to that, right? So the Rayleigh criterion for an optical microscope, provides the resolution limit, sort of the ability to distinguish two closely spaced absorbing or scattering objects, and the fundamental result that comes from that analysis is that in a light microscope, you can't resolve two closely spaced objects when they get closer than half of the wavelength apart. So the distance between the two objects become unresolvable if their distance is less than half of the wavelength you are using to image them. And so optical light has wavelengths in the hundreds of nanometers, right? So the typical range given for the optical spectrum is between 400 and 750 nanometers, and so that sets kind of a lower limit on spatial resolution for 400 nanometer light to be about 200 nanometers or so. And so just from that kind of simple description, you have a understanding of why there's no possibility of making atomic resolution images of materials with any sort of optical technique, right? So this Rayleigh resolution requirement kind of forbids it. The maximum spatial resolution you can expect from a light microscope is on the hundreds of nanometers length scale. I didn't talk about this in my seminar today, but put into the same context. Okay, the question is, why can you see atoms and resolve their spacing using electron beams rather than light beams? And the answer to that question has to do about the relevant wavelength of the electrons in the electron beam. And I think many people who are tuning in today will have heard of the de Broglie wavelength of matter waves. So, electrons are a massive particle, okay, but they're a quantum mechanical object. So they have an effective wavelength, which is given by the de Broglie equation, and so the effective wavelength of electrons in the electron beams that I was talking to you today about, ones that have a kinetic energy on the order of 100 kilovolts or so, the de Broglie wavelength, the effective wavelength of the electrons in that beam, are on the order of 3 picometers. Okay, so that's 100 times smaller. Okay, between 100 and 1,000 times smaller than the interatomic separations that are actually present in materials. Okay, and so that's the primary difference between electron microscopy and light microscopy from the perspective of spatial resolution, is because the electron beam, the effective wavelength of the electrons in the electron beam, okay, are sub-angstrom in the case of electrons, and hundreds of nanometers in the case of visible light. And so that makes electrons sensitive to interatomic separations and light microscopy insensitive to interatomic separations. I hope that answers your question.
Thank you, sir. And this may be the last question. What is the pump probe microscopy?
Right. Okay. So the pump probe microscopy is the experimental modality. So it's the experimental technique that allows you to actually reproducibly and reliably initiate dynamical processes in materials, and then to make these electron scattering images of the structural rearrangements that occur following photoexcitation. Okay, so if you want to be able to study a dynamical process, you kind of have a couple of options available to you, sort of a priori, right? You could hope to imagine realizing an instrument that was capable of, was basically the equivalent of a movie camera, but for molecules, right? That we could just film the structural dynamics in a molecule or a material as it occurred in its totality from start to finish. It turns out that that's not possible to do for all sorts of technical reasons. There's no such microscope that one can produce that allows you to make the direct equivalent of a movie camera, but for molecular rearrangements. That's not possible. So given that that's not possible, we have to be able to reproducibly and reliably both initiate and follow the atomic level structural dynamics that are occurring in the material. And the way that we do that is through this technique of pump probe microscopy, right? So the pump, okay, the laser excitation reliably and on a shot-to-shot basis yields the same structural dynamics result, right? That's the important thing. So optical excitation of the material yields the same dynamical pathway each time you excite the sample, right? And so that laser excitation both starts the dynamics, right? Okay, so that laser excitation starts the dynamics. So that does two things, right? It tells you when the dynamics start, right? So it gives you the starter's pistol for the dynamical evolution of the material, and does so in a reliable and robust way. So the laser excitation starts the dynamics, and the electron probe, okay, that follows that laser excitation probes the dynamics, right? So that's why we call it pump probe. Okay, the laser excitation pumps or stimulates the structural dynamics, and the electron pulse probes the result. Okay, the probe tells you about how the atomic arrangement inside of the material changed as a function of time. And in practice, you need to redo that many times for different time delays between the laser excitation and the probe, right? To get a full picture of the dynamics in the same way that I showed you those Muybridge images, right? The full picture of how the horse gallops is not contained in any one of those frames, right? The full picture of how the horse gallops is only contained in the sequence of frames, and by comparing how the different frames relate to the other frames in the sequence. And so the pump probe experiment gives you access to exactly that, right? For each setting of the time delay between the laser excitation and probe, you're only looking at one instantaneous configuration, right? But by sequencing these different frames together at different pump probe time delays, and given that the material dynamics are repeatable and reproducible on a shot-to-shot basis, laser pulse after laser pulse, you can sequence these pump probe frames together, okay, to obtain the same kind of picture that I showed you that Muybridge got for the horse galloping. That's the idea. So for one pump probe delay, you only get one of those shots, and then you redo that experiment over and over again for different time delays between pump and probe, and that allows you to get a different instantaneous image. And together, what you have is effectively like one of those flip book movies of the structural evolution of the material that is stimulated through photoexcitation.
Sir, for your wonderful presentation and discussion session. So as we have already told you that the main aim of our program is to motivate our students, and I think many of our students have motivated by your excellent lectures, sir. So
Great.
Thanks again for giving us this opportunity. And as we have discussed earlier that I'll definitely invite you for a face-to-face session. That will be very exciting for our student.
Great.
So
Well,
thanks.
this was a real pleasure. I was happy to be here today, and I hope, as you said, I was motivating and interesting and told you something that you didn't know about before.
Okay, sir.
Okay.
So bye for today. Have a nice day, sir.